Any given moment, trillions of cells are working and collaborating to keep your body healthy and functioning. Each cell in itself is like a tiny world, traditionally pictured as a smooth globe. In reality, though, it is more like a dense forest, with trees of all shapes and sizes, concealing countless unsolved mysteries. Instead of trees, this forest is made up of complex sugars, or Glycans, as they are scientifically known. Simple sugars, such as glucose, are known to all of us as an important source of energy. But did you know how essential Glycans are in making our cells work?
After 3 billion years of evolution, not a single life form has emerged that doesn't cover itself with a dense coat of glycans. With their branched structures, glycans are far more complex than proteins or DNA. Glycans are involved in nearly every biological process that takes place in our bodies, playing a central role in the communication between cells and the outside world. They are also involved in every major disease that afflicts mankind. The flu virus, for instance, infects us by taking advantage of specific glycans that cover the cells of our lungs. And in cases where healthy cells become cancerous, this is often accompanied by alterations in the glycan coat. These changes help cancer cells spread to other parts of the body by making them invisible to our immune system.
At the very origins of life, when the world was created, first core components of the living appeared. Glycans. Glycans are sophisticated sugar chains playing a key role in every aspect of life. Glycans provide energy to the cell, structure DNA. They shape and direct proteins and even determine blood types. Glycans are essential to cellular communication. Glycans are youth keys.
Secretory and membrane proteins are often post-translationally modified with sugar chains called glycans. Glycans are essential for the stability and function of the mature protein. They affect enzymatic activity, half-life, and receptor binding, among other biological processes.
Normal cells perform many functions essential to maintaining good health. They must be able to take in nutrition and eliminate waste or toxins. Normal cells must defend themselves from intruders like unfriendly bacteria, fungi, viruses, and free radicals, and repair the damage done by them. In addition, cells must work together as a team, communicating with other cells to achieve optimal organ and immune system function. Research has already proven our cells need certain essential nutrients, 26 different vitamins, 72 or more trace minerals, numerous fatty acids, and amino acids. Since our bodies don't produce these, we must get them from our diet. Recently, scientists have discovered that there are at least 8 sugars, called monosaccharides, that our cells also need for optimal function. Inside cells, these sugars combine with protein strands and form complex structures called glycoforms. The exterior of a healthy cell is covered with a dense forest of these glycoforms. Glycoforms can exchange infinite combinations of information depending on their shape and charge. Glycoforms are present on the surface of all cells and actually prevent viruses, bacteria, and other enemy cells from entering a healthy cell by blocking the entrance to the cell's surface. Glycoforms can hold on to an enemy cell until an immune system cell can arrive to destroy it. Healthy immune system cells can also accurately identify normal cells and not interfere with their function. Cells covered with glycoforms enable clear and accurate cell-to-cell and system-to-system communication, creating an information superhighway within the body. Glycoforms play a vital role in every human physiological process, immune system response, tissue regeneration, cell replication, growth, and structural stability. They are even responsible for the attraction of sperm cells to an egg cell surface to foster fertilization.
Healthy cells don't function optimally without glyconutrients. That's all there is to it.
We now know how important these 8 sugars are to human health. The problem is, only 2, glucose and galactose, are readily available in our modern diets. A fraction of these other 6 sugars can be gotten through diet, but modern agricultural methods, food processing, widespread use of toxic pesticides and chemical fertilizers have all but eliminated them from our food supply. Without vital sugars and a variety of other micronutrients, cells cannot create glycoforms. The result is a breakdown in cellular communication and immune system response. Depleted cells are unable to defend themselves effectively, so cell structures can be damaged and are not effective in healing and regenerating. In this weakened state, cells cannot communicate effectively, compromising the body's ability to identify problems. They are also unable to destroy pathogens such as viruses, bacteria, and fungi, and cannot identify mutated cells so the immune system can eliminate them. For example, when a person suffers from an autoimmune disorder, their compromised immune system may respond inappropriately and treat altered normal cells as intruders and destroy them. It may allow mutated cells to proliferate, or the immune system may not respond at all, leaving the body extremely vulnerable to many types of infectious agents.
Contact. This is the moment when infection begins. How does infection happen? You see the tiny shapes there on the virus particle's surface? They seem like random blobs. In reality, they are tiny keys, proteins that have evolved to the perfect shape and size, enabling them to unlock these receptors on the surface of this lung cell. Like a smile and a handshake, the virus deceives this lung cell and is welcomed in, where it will command the cell to make more virus particles. Every cell in your body is a tiny factory, constantly making proteins. Your genetic code, or DNA, directs every activity. Protein-making machinery in the cell reads the cell's own DNA and makes sure the cell proteins go where they're needed. Viruses have their own genetic code, but they don't have the machinery to copy their DNA. So viruses need your cell's machinery to reproduce themselves. A virus starts this process by using the keys on its surface to gain entry. Like a Trojan horse, it enters the cell. The virus releases its own DNA into the unsuspecting cell and hijacks the protein-making machinery. Now, this vital cell function becomes an assembly line for new virus particles. No longer in service to your body, the cell now serves the virus, manufacturing thousands of new virus particles which are released to enslave more cells, thousands and thousands more.
Scientists believe that influenza virus is spread from person to person through exposure to large respiratory droplets, direct contact, or airborne dispersal. Infection takes place mainly in the respiratory tract. Infection begins with attachment of virus proteins to a receptor on the surface of the host cell. The virus is then taken in the cell by receptor-mediated endocytosis. As the virus enters the cell, the virus is internalized in a membrane-bound capture vesicle that carries the viral core. The vesicle is transported on microtubules inside the cell by host proteins called kinesins. During transport, the membrane of the vesicle fuses with the membrane of the virus, and the capsid undergoes uncoating. The viral core RNA and proteins are then released into the cytoplasm, where they are guided by host proteins to the nucleus of the host cell. At the nuclear membrane, the viral core uses host protein channels to enter. Inside the nucleus, cell machinery is utilized by the virus to replicate the viral genome and make messenger RNA, mRNA. Some viral mRNA exits the nucleus to exploit cellular ribosomes to direct synthesis of viral proteins. Viral proteins go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface. Viral surface proteins are made and processed in the cytoplasm and also travel to the cell surface, where they combine with the encapsulated nucleoproteins to form progeny viruses, which depart from the cell by budding. The virion now goes on to infect other cells.
Basics of the immune system. The immune system, one of the most complex parts of the human body. An intricate network of cells and molecules evolved over the course of hundreds of thousands of years to protect us from the dangers of the outside environment. So how does it work? First, detect the threat, summon help, and launch a counterattack. When a virus infiltrates a cell, for example a cell lining our airway, the cell detects this as foreign and produces cytokines, messenger proteins, to summon help via phagocytes, a type of white blood cell, and also help to contain the local spread of the virus from cell to cell, typically the role of interferons. Specialized white blood cells called phagocytes try to destroy the intruding virus by literally eating them up. These first responders show up within minutes and make up the innate immune system. It is fast to respond, but not precise, and it does its best to shut down an infection as soon as possible. The adaptive immune system is highly specialized, but much slower. Dendritic cells collect small fragments of the virus left over from the battles of the innate immune system and ferry these to our lymph nodes, where highly specialized white blood cells called T cells are waiting. T cells are pre-programmed soldiers, each one trained to target a specific invader. When a dendritic cell carrying a specific type of virus fragment shows up, the relevant T cell will divide many times to produce an army of clones. There are two main types of T cells. Cytotoxic T cells recognize and destroy infected cells that harbor the virus, and helper T cells boost the immune response by activating B cells to produce antibodies. Antibodies are specific proteins that block the structures that viruses use to latch onto and enter our cells. Antibodies also mop up the viruses that are floating around within our bodies. Although T cells and B cells can take several days to move into action, they are incredibly precise. And they remember. After the virus has been neutralized and the army stands down, most specific T cells and B cells die off, but a small fraction stay alert, patrolling the bloodstream and lymph nodes for repeat attacks. If the same virus attacks our cells again, these memory cells spring into action, activating the power of the adaptive immune system without the days-long startup.
The complement system is an essential and powerful part of our immune defense system. Its main function is to coordinate the destruction and removal of infectious organisms, known as pathogens, or damaged cells in our body that need to be replaced. The complement system consists of a cascade of proteins, which activate each other by cleavage, the process of splitting each protein one by one. The complement system may be activated by 3 different pathways: the alternative, classical, and lectin pathways that all run through complement C3, the central protein in the cascade. C3 serves as the master switch, controlling all downstream effectors that ultimately cause destruction of the cell. The alternative pathway is spontaneously activated through direct binding of complement to a pathogen or damaged host cell to help the body clear away debris. The classical pathway is activated when an antibody, a protein produced by our immune system, recognizes and binds to a component on the surface of a foreign cell, forming an immune complex, starting an immune response that targets the pathogen for destruction. The lectin pathway is activated by natural molecules in our body that recognize sugars at the surface of infectious organisms. In situations where the complement system becomes overactivated, it results in immune attack, the destruction of healthy cells, and ultimately causes illness to the person.
Here, influenza viruses specifically attach to the surface of the epithelial cells. The viral membrane envelope contains the neuraminidase protein Na, important for the efficient release of newly produced viruses. The M2 ion channel promotes viral structural changes during cellular entry, and the influenza hemagglutinin protein HA, the key player for viral internalization, which facilitates viral binding to sialic acid-decorated receptors and subsequently viral fusion. Proteolytic cleavage by host enzymes is critical for the activation of the HA trimers. Soluble or cell-bound host proteases cleave the precursor HA molecules into 2 parts, HA1 and HA2. Influenza virus particles are internalized into early endosomes by clathrin-mediated endocytosis. In late endosomes, the pH drops, triggering the conformational change of the cleaved HA molecules. HA1 opens up and allows HA2 to form a triple alpha helix bundle, which extends towards the endosomal membrane. Once the fusion peptides are anchored in the endosomal membrane, the whole molecule can fold back, allowing the fusion of the viral and endosomal membranes. After fusion, the viral genome can be released into the cytosol. The 8 viral RNA segments make their way into the cell nucleus, and the production of the new virus begins. Just hours after the initial infection, thousands of new viruses bud off the cell surface and infect neighboring cells.